Okay, good morning, everyone. We have today a presentation by Lean, and she will start now, and I think it's going to take about 30 minutes. Lean, please go ahead, introduce yourself, and go ahead and start, please.
Okay, hello, my name is Lean Ahmed. Good morning, everyone. I hope you're all good this morning. Today, I'll actually be talking about a research that I participated in last year in my university's research center called Phenotyping Arabidopsis thaliana under nitrogen and salt stress conditions. It sounds complicated now, but I promise I'll be getting into more detail on what the research was actually about. And about how the experiment was conducted and so on. But before I get into it, I just want to give like a quick disclaimer that I'm aware this topic is not of everyone's interest. I just hope you guys give it a shot and listen, as I think awareness on environmental topics is very important for everyone. Okay, first I want to just go through the agenda, so how this presentation is going to go. I'm going to introduce the research, obviously, in the beginning, kind of define a couple of terminologies that will be used to understand this research. And then I will be talking about nitrogen and salt stress, which is the stressful conditions that we put our plants under. It was nitrogen deficiency and excess of nitrogen. I'll be getting into more details about it. And then I'm going to be talking about the importance of the research. So basically, why did the professor that I worked with fund this research and this experiment? Why does it matter to plant some plants and put it underneath specific environments? And then I'm going to be talking about the materials and methods. The materials and methods for every experiment, for every research, it's a very important part in the lab report after you're done with the experiment to kind of list all the tools that were used to make this experiment possible. And then I'm going to be talking about research data results, so just the outcomes of the experiment. It comes in graph form, so I'll show the graphs, and I will kind of summarize the research in the end. And then you guys can ask, there's a Q&A portion where you can ask any questions that you'd like. So before I move on, I want everyone to look to the picture to the right of the slide. This basically visually summarizes the research. To explain more, both rows of the plants it's the same exact plant, but in this case, the top row was subjected to more of a specific nutrient. It's whatever nutrient, it's not nitrogen, but it has more nutrient, and you can see it looks different. The phenotype looks different. And you can see like specific nutrient solutions can affect the way the physical appearances look, and you can translate that into graphs later on. Okay, so before we get started, I want everyone to kind of have like a basic knowledge and an understanding of the following terminologies, which is genotype and phenotype. So to the left, genotype is just basically all the genetic information and the DNA that's in our bodies and in every organism's body. This genetic information can later be coded to give out a specific phenotype. This is why we all look different. So we all have different genetic information, and it codes for a specific look, or it codes for a specific phenotype. And this research basically talks about the phenotype, the way that the physical appearance looks like after being affected by specific environments. Okay, so now to introduce the research a bit more, get into more details. Now that we know what a phenotype is, which is the physical appearance, phenotyping is the process of observing and measuring the physical, biochemical, and behavioral traits of an organism. So basically, this process aids scientists in the identification of genes that are involved in the stress responses. I'll be getting into more details about it. I don't want to get into too much of details here, but I really want to talk about nitrogen, as it is as I previously mentioned, it is the component that we kind of manipulated in the experiment. Nitrogen is a component found inside the chlorophyll that enables plant bodies to capture sunlight for photosynthesis. To put it in more basic terms, nitrogen is a nutrient, and you can find it in the chlorophyll, which is the green pigment that basically makes the plant appear to be green. So it's the pigment, but it also is the function, it has a function, a very important function in plants, to capture sunlight and use it for photosynthesis, which is, as we all know, how plants make their food. So this nitrogen is a nutrient that can be absorbed from the soil and basically optimizes photosynthesis for the plants. So basically, and in other words, having more nitrogen will increase the chlorophyll, and this is better for the plant. Next, for the model plants, actually, what is a model plant? Model plants are species of plants that they're commonly used for experimental purposes because they grow fast. So you can get data back fast and in shorter times. And these findings that are used in these simple model plants could later be applied on a larger scale and on more complicated plant species, let's say on a big crop field, for example, which has a big effect. So it might seem like, oh, it's just a small model plant, but no, the findings from these experiments can be used on larger scales. And the model plant that we used in my research was the Arabidopsis thaliana model plant. So that's what this long word means. It's the plant that was used in the experiment. And the last bullet point says that we observed 24 different genotypes of Arabidopsis thaliana. I will switch the slide to kind of hopefully get it to you more clearly. But basically this model plant has 24 different genotypes. So 24 different variations of its genetic information. So it has 24 different species basically. And in each cell here, you can see the different names of every single genotype. And we actually applied the nitrogen stressors on every single genotype, because if the genetic information is different, then the phenotype in the end will also look different. And this is what was important for my professor. She really wanted the phenotyping portion of this experiment. So for the next up, for materials and methods, I have separated into two parts. During the experiment methods, so basically when we were actually conditioning the plants to specific environments. And the second portion is after the course of the experiment, so how we collected our data and how we were able to reach to specific graphs that I will be showing later on. So for conditioning the plants, the first thing was Hoagland solution preparations. So this is the most crucial part, because we are supposed to be very careful when we are preparing the solutions that we are watering the plants in. So it's basically the nitrogen amounts and so and so. I'll also be showing it in a bit. But if you look to the picture on the right that I've attached, it's a real picture from when I was doing the experiment. You can see the solution in the jar. And the machine that's actually inserted in the jar is a pH meter. It calculates the pH of the solution. So we were very careful with everything. This is actually a very, very expensive machine. It's very expensive. And there's only one in our university research center. And like even when we're cleaning it after with the tissues and stuff, we need to be very careful because it might scratch the glass, and that would cost a lot. But next up, we have the climate cabinet. I also have a picture of the climate cabinet on the next slide. But the climate cabinet, it's like a chamber. You can insert the trays of your plants or whatever your experiment is about, and you can set this chamber to any temperature you'd like. So whatever heat you'd like, you can make it as cold as you want, as humid as you want. And here I've written the exact measurements that we put our plants underneath, like what climate we subjected our plants to. Now for the data collection portion of the methods, the first one was UV-VIS spectroscopy. This method basically records the chlorophyll content of the plants. So when the plants have grown, let's say it was after 4 months, we already saw like plants growing. We collected them, we harvested them, and we cut the leaves. And we put them under UV-VIS spectroscopy to count the amount of chlorophyll inside. So as I mentioned before, the more chlorophyll we have, that means our plant is happy. basically. It's producing green pigment, it's producing the chlorophyll. It has more chlorophyll. That means it's making more food, and photosynthesis is working better. And we have certain plants that actually looked purple. It had a purple pigment. Some had yellow pigment. That means there's no chlorophyll at all. And that means the plant basically did not enjoy the environment that it's in. And that helped us with our data when we were collecting it. And lastly was the drying chamber. The drying chamber, after like cutting the leaves and everything, we measured them, their fresh weights. We measured their fresh weight right after cutting them. But then after we put it in a drying chamber to dry out the leaves and measure the dry weights also. So both data was important for us, the dry weights and the fresh weights. So here are more pictures that I included from the experiment time. So to the left, it's just the weighing scales that I was talking about just now. So we measured them fresh when we were cutting them. We measured them fresh first, and we put in drying chamber, and we put it back on the weighing scale, and we measured the dry weights. And then to your right, you have the climate cabinet. So it's bigger than what it appears to be on the screen, actually. It's just very, very big. It's like a wall. And you open this chamber, set it to any climate that you want, and you close it carefully. And that's the growth chamber. Okay, so for nitrogen and salt stress conditions, I want to go quickly over it. As I said, first of all, we germinated the soil. So we planted the seeds of the Arabidopsis thaliana, and we put them into two different batches of stress conditioning. So the first batch was a batch that had excess of nitrogen in the soil. So it had more, like when we were preparing the Hoagland solutions, as you can see to the right, these are the exact measurements. But when we were preparing them, we put an excess of nitrogen to see how the plant would react. Would it like it? Would it not? And then we have the deficiency. We have the second batch, which was deficient in nitrogen. So it barely had any nitrogen. So if you look to the chart on the right, the white column, it's the 8 millimolar of nitrogen solution. So it had an excess of nitrogen. And then you have the 1.6 millimolar, which had the deficiency of nitrogen in the soil. And we wanted to see the results. How would the plant appear to be? How would the phenotype be affected by such environments? Okay, so I want to discuss why this research might be crucial. As I said, like, you'd look at the plants, you're like, okay, why are they planting these? Why is it important? What's the purpose? The first thing is environmental concern. So let's say these environmental conditions actually started occurring in our real life. We all know that pollution, we're facing pollution now, we're going to face it in the future, even climate change. So let's say salinity and nitrogen pollution. Let's take that as an example because this is what the research is about. If this were to occur in our environment on a larger scale, let's say on a big cropland, what would we do then? Like we need to do these environmental conditions on smaller plants so that if it occurs in that environment, we can have better environmental management and we can be able to control the damage, basically. And then the next thing I wrote here was the crop manipulation. So when we do these experiments on smaller plants, we're able to see how this plant responds as a stress, and then we can develop a new genetic variation to make this crop more tolerant to the stress. So basically what researchers do is they do this on a model plant, and they can pinpoint exactly what gene caused the plant to stress and look different as a phenotype, and they can genetically modify it. And if that were to happen on a larger scale, in like a cropland, let's say, they can easily manipulate all the plants or the crops to be more tolerant to these stressful conditions. And identification of stress responsive genes, it's repeating itself. It's the same thing over and over again. So these genes can then be further studied and potentially manipulated to improve stress tolerance in crops. So the research data and results, I thought I'd share like a bit of the plants that we were doing. Actually, this is just a small portion of it. It was a lot. We had a lot of plants and the experiment, but basically these are like 3 rows. The row to the most left is the control group. As everyone knows, in every experiment, we need to have a control group. The control group means it was perfect conditions. It's the normal conditions, assuming there is no pollution, assuming we have perfect soil, perfect amount of water, perfect amount of sunlight. That's the row on the left. And the one in the middle is actually the nitrogen excess conditioning. We were actually very surprised. As you can see, the row of plants in the middle is the smallest. We got the least results, and we actually thought that having an excess of nitrogen would make it big anyways, because we need nitrogen. But no, having an excess of nitrogen actually caused the plant to look the most different. And then you have the column to your right. The most right was the nitrogen deficient conditioning. So it had less nitrogen, but surprisingly, it actually grew bigger than having too much nitrogen. So these were our results. As you can see, these are the graphs that we were able to collect after we collected the data from the fresh and dry weights. If you look at the x-axis, these are all the 24 genotypes that I talked about or I mentioned before. Every single one of them is a name for a genotype of the Arabidopsis thaliana model plant. And the bar columns, the yellow ones are the nitrogen deficient conditionings. As you can see, the yellow ones are always longer than the red ones, which are the nitrogen excess conditions, which again, it surprised us, but those were the results, both for dry and fresh weights. Okay, so in summary, the alteration that is done by subjecting the model plant to different salt and nitrogen conditions obviously resulted in several different differences in their growth rate and the way that they looked. So I'm going to show a little video here to kind of explain a bit what I'm talking about. As you can see here, some plants are bigger. Some plants appear to be purple, which is the purple pigment. It's called anthocyanin. That means the plant is not liking the environment, obviously because there's no production of chlorophyll. And I want to show you guys, see here to the top, there's barely any plants. This could actually be either a fault from us when we were germinating the seeds and putting it in the soil. So it could be our mistake, or it could just, again, be just the plants didn't like the environment, it couldn't possibly grow, and so on. So again, I just want to remind you guys that these tests are very important. It looks very silly maybe, but these tests are actually very important because they can be carried out as preparation for future environmental crises. So if any type of pollution happens, let's say nitrogen in this case, we would know exactly what genes cause the stress to the plant, and they can manipulate the plants. Okay, I'm done. I hope you enjoyed my presentation. If anyone would like